as we discuss, the DNA, foreign DNA is introduced into the host cell for cloning, or that may be cloned in PCR method. PCR based cloning also done. However, the motto is to obtain the foreign gene product, and they have to be processed and packaged and marketed. Our motto is to obtain the product, then it has to be processed for marketing after getting packaged. So, how it is done? Obtaining the foreign gene products, the recombinant DNA or DNA, when the passenger DNA is incorporated, joined with the vector DNA, rDNA or R plasmid are produced, and they are introduced into a host cell, that is a bacterial cell or the target cell, plant cell or animal cell. This aligned DNA then multiplied within this host cell or plant cell or animal cell, okay, in the culture medium, or when it is introduced into the bacterial cell and it is multiplied due to the multiplication of cell multiplication of the plasmid. Even if they are the part of remain part of the plant cell or animal cell, they are being cultured. These plant or animal cells are cultured. Thus, the ln DNA, foreign DNA is also multiplied. Then, in optimized condition, the condition required for the expression of the gene, expression of the genes. In optimized condition, the target proteins are expressed. That has to be maintained in large scale in the industries or in small scale in the laboratories for research purpose. Okay, in the laboratories, in small scale it is done. The proteins are extracted from these cultured cells. Cells are cultured. So, from the cultured cells, the proteins are extracted, okay, from the cultured cell. Then products, they are purified. They have to be purified by using the different techniques, by using the bioreactors. See the bioreactors here. Bioreactors, this is simple stirred tank bioreactor or spark stirred tank bioreactor. Okay, here what happens? Stirring, moving means rotating. This motor is here, that the middle central rod to the central rod blades are attached. Okay, the culture broth is here, means the culture in the medium the cells are cultured. That culture broth means along with the medium, along with the cultured cells or the cultured microorganisms, they are present over here. And this is steam is provided from this side. Sterilization, steam for sterilization means that to keep it in microbe free, to keep this internal environment. This is the closed bioreactor, okay, to keep it the microbe free, okay, the contamination free air, that is steam for sterilization. Acid or base is added from this side for maintaining to control the pH. Then the sterile air means contamination free air, okay, that is released from this side. However, the product, when they are here, this is spin stirred. By this what happens? They are processed, and products are that are processed by using this stirring technique within the closed tank, or it can also be processed by using the spark stirred tank bioreactor. Here what happens? This is the open tank. The sparked means sprinkled water is applied to create the bubbles or air spaces for more aeration. What happens? The aeration means it keeps the microbes on aerobic respiration. Okay, with the oxygen is supplied to keep the bacteria or microbes which are used as a host cell, they can be under the aerobic respiration. Okay, and then when the processed proteins, okay, the products are collected from these bioreactors, and then these synthesized products, these products are collected, extracted. What happens? The target proteins, these are extracted from the cultured cells by using the bioreactors. The products now collected, the synthesized products has to be processed by the process of separation and purification. They have to be, the product has to be separated from other such products, but our desired products we have to separate from other such cellular products. So the cells are producing different proteins, but which protein we need, we have to separate it. So separation and purification, the protein has to be purified. Suppose we need the human that insulin, that is also protein, or any other drug, the growth hormone, these are different proteins and finished products by this, and this is called downstream processing. The separation and purification, the method of separation and purification, this is called downstream processing. This is only called downstream processing or final processing, where the finished products are availed. We can get the finished products. This is the final processing. That's why downstream processing. Here in bioreactor, we processed it to extract the protein from the cultured cells. Okay, but again it has to be purified and formulation with the suitable preservatives, because it has to be marketed. So preservatives are applied to these proteins, suitable preservatives. If these are the drugs, medicines, then the clinical trial is done before marketing. Then quality control is strict, quality control is done. The strict quality control testing, strict quality control testing is done, then packaging, then marketing. This is all. So what we discuss in this long question, the steps of recombinant DNA technology, what we discuss, the different steps. The step starts with the isolation of the passenger or vector and passenger and vector DNA. Then passenger and vector DNA, they are cleaved. So next step is cleaving of the DNA, that is passenger and vector DNA. Then the separation of the different fragments of different size of that the passenger DNA, not of the vector DNA. Vector DNA is the plasmid, it is cut, cleaved at a particular restriction site. That's all. There the insertion of the passenger DNA has to be done. So only the separation of passenger DNA is done. Passenger DNA fragments are done by using the gel electrophoresis method. For the separation of the DNA fragments, the agar gel is used. Then the blotting method, that is the transfer of the separated DNA fragments onto the nitrocellulose membrane or the nylon membrane. Then what is collected? Our digested vector is ready. Now that cleaved plasmid, okay, is ready, and along with to that vector, the DNA, the alien DNA or the foreign DNA fragment is joined by using the ligase enzyme by incubating those both of these passenger and vector DNA along with the ligase. So our DNA is ready, our DNA, the chimera, our plasmid is ready. Then it is introduced into the host cell. Next step is introducing the rDNA, recombinant DNA, into the host cell for cloning or into the target cell for the expression. Introducing into a cell, bacterial cell or plant cell or animal cell. Generally it is introduced into the bacterial cell for the host cell based cloning. Then cloning is done either in the host cell based cloning or the PCR method. That PCR based cloning is done, polymerase chain reaction, by using a thermocycler. Okay, it is cloned, then the part, that's a step comes for the obtaining the foreign gene product. Okay, that by using the different technique, by using the bioreactor, stirred tank bioreactor, okay, that it maybe uses the simple stirred tank or the sparge stirred tank is used to obtain the product, means extract, to extract the that products from the cultured cells. Then they are processed. Processing is done to just separate and purify, separate the desired protein, desired product from other products of the cell. Then it is purified. That process again, formulation with the suitable preservatives. Then their quality checking, quality control testing is done. Then they are packed and sold in the market. They are sent to the market. Okay, this is all about the recombinant DNA technology. Steps of recombinant DNA technology. Each step is also important for 2.5 short note. And PCR itself, that's why PCR, the cloning is given in separately, because PCR itself is important for the short note of long question. In the long question, 7 marks long question is there, 3.5 is there. For 3.5 it is important. So complete all this, practice all this. Okay, thank you.